Welcome back to Saving Sapiens. Today, we are very excited to welcome for a second visit Dr. Varun Dwaraka, the principal investigator and research director for Epigenetics at True Diagnostic, one of the leading companies working specifically in the science of the epigenetics of aging. Welcome, Dr. Dwaraka.
Thank you so much, Dr. Matt. Yeah, absolutely. I'm glad to be back. And I have to give kudos to you because you've had some fantastic individuals on the podcast recently, and I'm honored to kind of follow suit with them as well.
Thank you so much for making time. We're very keen on the constant science updates on a couple of fronts that have really kind of led much of the science and medicine news cycle, especially this past year. All of us are aware of the constant updates on the front of GLP-1 agonist drugs, the cutting edge new technology that is allowing the populations that frequently suffer from chronic disease to have a new alternative therapy for weight loss, for battling some of the traditionally non-pharmaceutically treatable chronic illnesses. But we don't usually see a crossover with epigenetics and aging. Can you catch us up on what has been happening and why this has become a huge and recurring news story?
Yeah, absolutely. I will probably start by saying I'm very surprised, equally as surprised as many people, about how this study was picked up, because just to kind of catch you up, this preprint that we put out was actually started off as kind of a question of whether semaglutide or any of these GLP-1 agonists that you can buy, like you can get prescribed or you can kind of get off the shelf, how do they affect this concept of epigenetic aging? I think in my first podcast, I really went through and described what is epigenetic aging? Why are we using DNA methylation as a conduit or a biomarker to look at this biological aging front? And so more recently in the last year, we've been applying a lot of these clocks in many different interventions. And so we actually had a collaborator of ours, Dr. Michael Corley from now UCSD, formerly of Cornell University. He came with this interesting data set, said, hey, we have these individuals that are HIV associated, that have HIV associated lipohypertrophy, and we have their PBMCs. One of the questions, and they were randomized, where one randomized group went into a placebo and the other randomized group went into Ozempic and I think Wegovy. I believe that name is used synonymously. And so we essentially just wanted to ask, is there a significant difference or change in their biological age as calculated as these DNA methylation clocks? And so we did the study, we reported it, put it out as a preprint, and then a lot of these viral news stories of, Ozempic or is able to decrease your biological age comes out. So to kind of maybe give a little bit of context, we didn't come in really just to investigate semaglutide. We wanted to see the responsiveness of these epigenetic clocks on semaglutide. And what was interesting, maybe just as a high level output, is that we saw significant decreases in the randomized group that had GLP-1 across multiple different clocks. This didn't just include your Horvath clock, your hand clock, but it included a lot of the pheno-age clock, Grimm age, Dunedin pace, a lot of these epigenetic clocks that are built on biological output. We saw significant decreases there, which were not observed in the placebo group. And so maybe kind of coming back to that initial question, yeah, it's interesting that we saw that deviations, and now the questions have been coming around as to why. What are the mechanisms?
Can you give us a little bit of a background primer on, for people who are not familiar with the concept of epigenetics, give us a little bit of the cliff notes on why epigenetics is something we would be measuring in the middle of a patient's life, as opposed to something like genetics, where doing a genotype on a patient as an infant versus an elderly individual would result in the same results, as opposed to epigenetics, where you're able to go in and take measurements before and after and during treatment initiation? What is the difference here for our audience?
Sure. Yeah. So when we think about genetics, this is kind of what you can look at when you're taking genetic testing. Formerly 23andMe was a huge commercial brand that looked at the genetic testing, and then you can also do this in your current practitioner's office. So when you think about genetics, you're thinking about the DNA molecules that make up your genome, or essentially the blueprint and how your cells are made up or how you are made up on a cellular level. Many of these nucleotides, these DNA bases and nucleotides, don't really change as you grow older. You have a fixed set of your DNA composition, and that's it. As cellular replication occurs or as you grow older, the same DNA nucleotides and the same genome, same DNA, how would you call the sequences, are essentially stagnant and static throughout.
And these are the genes that we're used to discussing that you get from your mom and your dad that you pass on to your children. These are the static genes that you actually have crossing generations.
Exactly. That DNA nucleotides can make up the genes that are actually being passed on generation by generation. One of the beautiful things that the cell has figured out is that, okay, even though I have the hardware or the DNA that's around, based on the interpretation of the environment, we can make changes, because as an organic being, you have to be resilient to the environment that you're around. And so rather than changing the DNA, which could cause deleterious changes or affect negatively what could happen, what the body has figured out, or what these cells have figured out, is, are there other biomolecules that you could use to mask, hide, or maybe express certain of these genes or these DNA regions? And that's where epigenetics really comes into play. I will have to say, epigenetics actually goes multiple facets. You can have the modulation of the proteins that bind the DNA. So this is kind of what we call as chromatin changes or histone modifications. You can have the physical biomolecules add onto the biochemical marks, add onto the DNA itself, which is DNA methylation, which is what we kind of focused on this study. Or you can have non-coding RNAs and other biomolecules that actually repress or express, open up the expression of those areas, where it's literally covering, hiding out, or opening up the areas of the DNA so that they are actually expressed. And so that's where epigenetics comes into play, is looking at the expression of the DNA nucleotides or the genes that make up the genome. Now, the reason why it's really interesting to look at your epigenetics is because, as you can kind of intuitively understand, as you're existing within the world, your body is responding to the different signals that are coming out that are extraneous. Like, for example, the pollution levels, the type of diets that you're eating, stress, things like that will modulate how your body or your cellular mechanisms are responding. Rather than changing the DNA, because DNA cannot change, why not interpret it different? And that's where your epigenetics are modulated throughout not only your life, but even within a couple of weeks. And that's where we as the True Diagnostic, the company that did a lot of the research along with Michael Corley from UCSD, we really specialize in how do you harness the epigenetics, make a test out of it, and then provide that information to really give the health understanding or the relationship to biological age based on the epigenetic biomarkers. These biomarkers that are actually more in line with your exposure or your exposome compared to something that's static like your genetics.
So a person who's exposed to a lot of environmental stressors, we can assume might go through epigenetic changes that are approximately equal to having been alive a few more years than a person who's exposed to fewer environmental stressors.
Those are the observations we're seeing right now, is that this is where the concept of an accelerated biological age or a decreased biological age really comes into play, especially from epigenetics, where we're seeing that if individuals are, let's say, maybe eating a diet that is causing a lot more inflammation, that actually translates a little bit to a higher biological age output, or just worse measures across many different epigenetic derived healthcare outputs. And this is kind of where, I think in popular culture and popular media, the individual like Brian Johnson is able to say, "Oh, I age like an 18-year-old, even though I'm 45." It's that concept that, by making those changes, it really does reflect on your epigenetics.
So one of the things that is kind of at the forefront of what I'm always wondering when new epigenetic association studies are published is whether we're able to kind of delineate between what is the causal driver and what is sort of a correlation. There's a very frequent objection that we see always anytime any new research is published, when people run to the comments and shout, correlation is not causation. And to some degree, that is true, but in some sense, all causation is initially discovered through correlation studies. The question that I'm left with is whether we have reason to think that a new drug is driving clinical outcomes that appear to reduce the physiological stress for the patient. Like, obviously, GLP-1s are enormously popular right now. Whether listeners have heard of it under names like the names like semaglutide or tirzepatide, or whether they're hearing names like Wegovy or Manjaro or Ozempic, the primary outcomes of weight loss, reduced, we think possibly reductions in cardiovascular mortality, all of these things are giving us reason to be unsurprised by the enormous clinical efficacy. And now I think the numbers are running at that it's probably going to approach $100 billion industry within a half a decade. But this opens up a whole new take for the rest of the population, is what we immediately want to ask, because it looks like either indirectly it's driving reduced epigenetic aging, or directly, do we have a way of actually distinguishing between those two possibilities?
That is a fantastic question, and it's a question that I think about a whole lot. I know Matt Kaberlein and Peter Tia have recently been discrediting a lot of these epigenetic clocks as maybe just research tools compared to actual clinical tools, and I think that fits within here. To be very frank with you, we are still going through and trying to tease out how much of this is correlation versus causation. Because in order to make a causation specific claim, you have to be able to modulate some of these biomarkers. For example, in genetic studies, they ablate or they remove or knock out the gene in question and then see what the outcome is. The problem with doing this in a epigenetic study is that, if you modulate the specific CPG site, typically these DNA methylation marks don't exist as a single entity. They are working in tandem with neighboring CPGs. And so in order to do this, let alone, I'm not even going to touch the whole human subject. Doing this in let's say a rat model or a mouse model is very difficult, because you don't know how much of a compensatory effect there is if you remove one CPG and affect the other. So as of now, I am going to lead on the side of saying a lot of these correlations that we're seeing tend to show that as the individuals are taking semaglutide, their DNA is modulating. That's for sure. We're seeing changes in the DNA methylation, which is then resulting in a lower epigenetic age as calculated from these epigenetic clocks. Now, but even with that said, I think we're getting closer to the causation, because there was a recent paper that actually used CRISPR dead Cas9. So this is the deactivated Cas9 protein, where you're not removing specific genetic locations, but it was designed to actually remove and add DNA methylation sites. And they were able to remove these DNA methylation sites specifically for these clocks. And they showed that the removal and addition of these particular DNA methylation sites that are, I think the one that they looked at was the Horvath clock. It actually increased and decreased the overall epigenetic age of that individual, of that, I think, cell line. This was done on a cell line study first. So I think we're entering the realm of the causation with this. But in order to do that for these individuals, we'd have to be able to show that in a cell line first and then mouse model, that semaglutide or affecting these DNA markers that are changing are modulating the epigenetic age. The reason why I still think that there's importance here is, you kind of mentioned it yourself, the correlation does imply some level, it can imply some level of causation that needs to be tested. That's the hypothetical question that now has to be tested. But a lot of these markers were developed with the mortality outputs and odds ratios, associations to disease in mind. So for example, some of the clocks that really struck us as incredible were dunedin, pace, and Grimage, the first version and the second version that were developed by Eve Horvath's group. The reason why I'm really touting these three is because the way that these were developed and the associations to disease, these tend to show strong signatures of mortality when you do these kind of like, associations tests to mortality risk, and also specific cardiovascular disease and diabetes risk. They are heavily associated, where increases in these clocks are associated with increases in mortality risk, cardiovascular disease, and diabetes. And though especially CVD and diabetes are very consistent with what semaglutide is supposed to be decreasing. And so that's why I think that there's a strong case to show that those changes that we're seeing are potentially showing beneficial outcomes for the semaglutide study that we did.
When discussing the sort of cutting edge medical genetics and epigenetics with a typical clinician, I think a knee-jerk reaction a lot of times is, hey, the patient feels better. They're finally in maybe a weight range or a sort of dietary intake that their body is happy with. The stress on their joints is improved, their mobility is improved, and all of these sort of clinical correlates of having the patient on this therapeutic are things that line up with them feeling younger. And it's fascinating that we can measure their epigenetic age. The hanging question with the correlation versus causation, of course, is if we're not yet at the place where we can propose a direct epigenetic modulation mechanism by which these GLPs are interacting with the epigenome, we almost have to ask the question, if a patient could achieve similar outcomes through perhaps dietary or lifestyle modification versus GLPs, is there going to be, in your prediction, a difference, or is the indirect causation of simply adjusting the physiology to these more optimal levels? Do we expect them to both have a similar outcome? And the reason why this is such an interesting question for me, for my patients, is ultimately both because we want to ask the question, how can we optimize your health span with or without the prescription therapeutic, but also because we want to know if we have somebody who's optimizing their lifestyle factors, should they also be on a GLP-1? Because if there is a direct mechanism, it would be pretty cool to know if we can further slow aging. Is this something that we can, is this something we could can look into? And I imagine a lot of the news cycle is actually circling around it because of this should we expand this to treat everybody?
I will say first, so this is the first study to even show that relationship between the epigenetic clocks and semaglutide. I am a strong proponent that in order to increase health span or to optimize health span, it has to come from lifestyle. A lot of these studies that we do, especially at True Diagnostic and with our collaborators, have really shown that the majority of these clocks really are optimized based on lifestyle changes. So I will probably be maybe screaming the same thing a lot of the physicians will be saying, don't just take semaglutide just because it decreases your biological age. It is a tool that should be compounded with lifestyle changes. But I think as we move forward, I do acknowledge that there are some individuals with pre-existing conditions where simple lifestyle change is not necessarily the only thing you can do. I've met so many individuals just in my personal life and, going to different conferences where, hey, I work out every day. I do all these things. Again, I'm not a licensed physician. I'm a scientist first and foremost. But, when I hear these stories, I can't help but stop thinking about the idea that even if you do everything like, working out, eating right, being on a caloric deficit, getting your 8 hours of sleep, there's still some pre-existing conditions that could be epigenetic and genetic in nature, right? There's still a correlation between genetic and epigenetics where even if you have certain SNPs that are not allowing your metabolism to be at the most optimal method, the epigenetics will work in tandem with those deleterious genetic outputs. And so I think for those individuals, that's where, the semaglutide could come in to help as a tool along with those lifestyle changes. And that's kind of where I'm existing in this news cycle. I've seen, just take, you just take Ozempic and you can decrease your biological age. And no, not necessarily true, because even in our study, the individuals that we looked at are specifically HIV positive and already show lipohypertrophy, which means that they're already comorbid. And so for those individuals, this semaglutide, along with, the additional changes that they possibly made in their life, did show a decrease. So if you fit that bill, then maybe, semaglutide is a good way. And one of the outcomes is going to be the change in that biological age. But I think for moving forward, to really address that question about, how does this work maybe with the average individual, the health optimized individual, we'll have to redo this on a healthier population, those without maybe existing comorbidities.
And that was going to be my very next question. Every single time there's a new claim to expand some pre-existing pharmaceutical to the general population, with the understanding that we are somehow pushing lifespan, the immediate question I always have is, has this been tested on, say, humans versus rodents? Or has this been tested, if it is in humans, has this been tested at the general population under a controlled study where the population that we're investigating isn't a population already suffering significant setbacks from a long-term illness? And to nail this down, for anybody who has seen the headlines that GLP-1 makes you younger, this study specifically was exclusively looking at patients who were HIV positive and had the lipohypertrophy already present.
And one of the things here too is that we're hoping that a lot of this generalizes. And that's kind of the next step. We've already been in contact with a few doctors that are already prescribing this and with a specific demographic. And actually, Michael Corley, we spoke recently, and he actually has a washout data too. So individuals 5 years down, it's not 5 years, 5 weeks down the line that have been off of semaglutide, just to maybe even look at the idea of what happens with the age when you get off. Like, does it kind of come back the same way as, for example, with weight and semaglutide, individuals who get off of semaglutide tend to get all that weight back because, one of the things that people think is that they haven't made lifestyle changes to really keep the weight off. So we're studying that. To your point of doing maybe another randomized control trial with healthy humans, that would be a perfect, complement to this. But again, getting the IRB for individuals who are already healthy, but then, convincing a board to say, hey, we should just put them on GLP-1. It's still early even with GLP-1 because we don't even know what the long-term effects are as well.
Which is something that is really, really hard to emphasize on a drug that is so incredibly popular. We do have some data again on patients who are diabetic, who have been on products like this for a very long time. And a lot of times, because we have good safety and efficacy data relative to the mortality and morbidity for an untreated person who also has diabetes, we tend to make a lot of our sort of safety decisions based on what is your mortality morbidity risk if you have untreated or undertreated diabetes versus if you take this. And the majority of the physicians that I've discussed GLPs with, when they discuss the safety and efficacy, are framing it within these long-term studies, oftentimes not explicitly cognizant of the fact that those safety parameters are actually established relative to not giving a diabetic a GLP-1. And so when you decide, hey, this is an acceptable risk profile, you're typically doing it based on the assumption that the person will decline with that chronic disease. And I think that is clouding some of the decisions that we have to make when we're thinking about arguments over whether metformin or rapamycin or a GLP-1 or some other therapeutic could, should, or would ever be rolled out at the population level for people who are not chronically ill from something.
Yeah,
agreed. Do you have an idea of the I guess the most effective way of how do we most effectively communicate that there isn't an escape route? Because I feel like a broken record player sometimes when I tell people you can't escape evolutionary contingency. And
Mm.
ultimately, our ancestors walked like 10 kilometers a day and ate 100 grams of fiber a day. And just because a drug has the ability to reduce something like a chronic illness, how do we best convey, as a person who's constantly in front of cameras and the media and everything, also, how do we convey that there isn't an escape button, whether it's GLP-1s or anything else?
I mean, I think you said it there, Matt. I mean, what you're doing right now is educating the masses. Like it does suck to be the person to just keep like, shouting, but I think reframing it in the sense of, how do we educate the individuals to be more, let's say, woke about these, stories that come out. One of the things, I guess I put it on Instagram was, really looking at how different periodicals show the data has been a huge thing that I've been trying to even tell amongst my friends because, like, hey, you were on the New York Post and you said that you could take semaglutide and it'll reduce your age. Like, how did you, like, that's so cool. And I'm like, whoa, 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 whoa. And then like, kind of go through versus like, New Scientist did a fantastic way of like, highlighting, hey, this was a HIV specific, cohort. They looked for 32 weeks. Yes, there was a placebo versus administration group, which is awesome. It was, randomized control, but like it actually went through and hopefully opened up those ideas to the general population so that they can also look back and say, okay, there's limitations, but they're still seeing something different. So I think education is going to be huge. And it's another reason why, even as a for-profit institute, we work very closely with researchers is because by publishing this information, what we're hoping is that we don't necessarily want to disseminate it just directly to the public. We would love to, but I'll be very honest, there's a couple of steps that have a gap between the general populace and, like, let's say a published, I don't know, Nature paper. We want to disseminate this to the individuals that are the healthcare practitioners, the doctors, the individuals who are having direct, let's say, contact with these patients so that they could read these papers and then understand, okay, how do you show the contingencies? So I think that that's the way that we've kind of come across or have been trying to go with disseminating this information. Because I think that the published work is, I mean, of course, the elephant in the room is this is still a preprint. So we are going through the peer review process, but hopefully we're really crossing our fingers. Once it gets the published state, then that's where we can then, send this to the right individuals so that they can communicate effectively and honestly.
The present era of rewarding clickbaity claims and extreme news headlines has made it almost an art to actually convey what real cutting edge science is for that particular week. Like, if you are in science and medicine and you follow Nature or JAMA or, the BMJ or anything like that, you're aware of what a normal peer-reviewed science headline looks like. But the percentage of us who are following the actual headlines as opposed to the number of clicks that the rest of the population is consuming makes it almost easier to have a viral story about,
Yeah.
alien UFO, something this, something that, or, magic pill makes you 10 years younger kind of thing, more than the sort of nuance of the science. So we really, really appreciate you taking the time to kind of slow things down, think about the hype versus the real science, and then kind of giving us that perspective on what the next steps could be for research, what kind of questions we should be asking, and the questions that have actually been answered thus far. Do you have any insights that you can share with us about any other either therapeutic, interventional, or lifestyle things that might be the cutting edge for sort of fighting the epigenetic aging front in the coming years outside of GLP-1s?
Absolutely. And actually, one of the reasons why we were really excited, and I think the field is generally excited about semaglutide, is the weight loss aspect. Weight loss kind of derived from caloric restriction. The reason why is because what we've seen in previous studies of RCTs with epigenetic clocks in particular is that if you do a 20, I believe it was a 25% reduction in caloric intake of your what your daily caloric intake should be. If you do a 25% reduction, almost after a year, you're actually seeing significant decreases in a lot of these biological age clocks. In addition, another study that we published about a year or so ago went viral, which was the omnivore versus vegan diet.
Mhm.
That was a RCT trial too. And while, there was all these claims of, oh, vegan diet is actually going to decrease your biological age in 8 weeks, which is what we saw in that group of individuals, what it actually kind of led us to think was this whole concept of caloric restriction, because one of the limitations of that study was the fact that we weren't able to control for that the caloric deficit that the vegans were undergoing, mainly because we wanted to do this in like a free living state. We didn't want to do feeding them like they were rats in a lab per se. And so it really comes down to this caloric restriction. It seems like what's underlying all of these different decreases and some other additional therapeutics seems like caloric restriction is having a huge impact on the changes in your epigenome that seem to be decreasing the clocks themselves. Now, there is a question whether the clocks are tuned to caloric restriction specifically, or if this is let's say a consequence of caloric restriction, or if it's just coincidence that it's happening.
And that's almost the same question. It's almost the same question with the GLP-1s. Is this downstream physiology driving the epigenetics?
Yeah. But at the same time, I mean, with other additional, like not even looking at epigenetics, but a lot of your HbA1cs, your clinical scores tend to improve with caloric restriction. So it seems like caloric restriction, and not even just intermittent fasting, but just generally existing in that caloric restriction state seems to have improvements not only on the epigenetics, but on the clinical side of things as well. And then obviously nutrient optimization. So there was a fantastic paper that came out by, I'm going to butcher the last name, Heinke Bischoff, that showed that omega-3 consumption by itself is able to decrease many of these biological clocks like gramage, phenoage. I believe they also looked at denine pace as well. So it's really coming down to, caloric restriction seeming to be the biggest, like the web that connects all of these improvements, but then also the nutrient optimization that we're seeing as well.
I'm so torn on both of these, because the moment that we have the nutrient optimization ones, they're super valid, super important, and super insightful about the sort of metabolomics that we need to be focusing on. And so they should certainly be funded, and we should pay careful attention to them. My instant concern pops up over the supplement industry, just because of how viciously they tend to jump on any of the nutrient optimization studies to start peddling some kind of supplement to meet some kind of deficit. So I'm curious to see how that plays out. The calorie restriction one is kind of this recurring theme that has gone from an observation principally in non-human studies initially. So for a bunch of years, it was really sort of tightly observed phenomenon in non-humans that matched a lot of the data that we had on actual wild studies of organisms who appear to have metabolically slowed lives. So if you follow things like the calorie intake for excessively long-lived organisms, whether it is, specific tortoise species that can live several centuries, whether it's some of the cold water shark species that live near the Arctic Circle, and their very minimal calorie intake and extremely slow metabolism and extremely long lifespans, we had probably at least 4 or 5 completely independent sort of biological reasons to think that these calorie reduced lives were somehow allowing aging to occur more slowly. And the sort of exciting takeaway of like, hey, maybe we can replicate this in humans is the biggest sort of almost more of a possibly both a biological question and a human psychology question, because the question is still out, just hands down. The question on whether calorie restriction works, I will be at the edge of my seat trying to follow what you guys find epigenetically. Even this is one of the few areas where, like, even if you do find that a significant calorie restriction actually does slow aging and it's confirmed to be a direct causation in humans, man, I genuinely think this might be the one discovery that would make possibly zero impact for at least for the population in the West, simply because the sort of sheer willpower based reduction of your calorie intake by 25% goes against every adaptation that we have for the last 300,000 years.
Right.
Every ancestral survivor who like survived to have a kid because like they scrapped for that 10 extra calories here and they found that one extra little pocket of seeds or rotting fruit or something that they were able to scarf down.
But the thing that you just mentioned right there, and I'm going to say I think because, again, I have to maybe spend a little bit more time on the caloric restriction, but I think that's the main thing, right? Because I think the standard American diet is inflating a lot of these calories based on the type of, the type of foods that are being made. For example, I completely agree. Like in the evolutionary context, we came from scavengers where we were going around following the herds of like woolly mammoths and whatever, trying to maintain a fuel source. But that fuel source was easier to digest. It was a lot more organic. There's a way that it was generated which was able to sustain us in a right way and also allow for satiation, but then also ease of caloric restriction. But I think that what I'm hoping that changes and that increases longevity is the way that convenience fast food that changes so that it kind of mimics a little bit more of the raw products, the natural products that really were existing out there. And I hate saying it this way.
No, that's a really good insight because the majority of our colleagues in the animal world, they do have to scrap and they have to eat almost all day, almost every day, because they're exclusively eating poorly digestible raw foods. And we unlocked cooking so we could cook the mammoth meat, which made it easier. But then this explosion in the last 50 years of hyper nutrient density, ultra sweetened, high fructose corn syrup, everything. You're absolutely right. But are you suggesting that this kind of insight wouldn't necessarily be for the individual patient, but more give us reason to perhaps shy away from ultra processed food? Is that kind of the pitch?
I think the pitch is to two folks. So for the patient, I think working with a registered dietitian, because again, as a company for True Diagnostic, that's why we exist. Is we don't really have a lay of claim in any of these different optimization techniques. What we want to provide are the tools so you can track it on a cellular level. And so if you're working with a registered dietitian, I think that's the best way. And then using some of these tools to really look at what are those nutrient changes, are you optimizing for that? And it doesn't just have to be epigenetics. Use the clinical markers as well. I think that it gives you an encompassing thing. But then maybe my own personal view is it really got me thinking about shifting the mindset of convenience foods. So like the PepsiCos of the world, the Frito-Lays, like all of these companies that are not there to just generate food, they're generating convenient food. So how do you improve the composition of the convenient food so that even for an individual that is not able to make, a certain salary a year can still have that convenient food available. Mm-hmm. And I think that's a fight that I don't even know if I have even any lay of claim, but it's got me thinking after looking at a lot of these studies and really, discussing even with, a lot of the dietitians and also the nutrient researchers of the world.
Yeah, we can imagine a world where the options to every patient, whether they're rushed or not, whether they have the finances or not, would not push them past that calorie threshold. And I think we can all agree that this would be an optimized scenario relative to what patients are facing in the West. And that's kind of our ultimate goal, right? We want to give everybody all the tools that they can to maximize their health span and to do so in a way that is not just internet hype, but is based on real science. So thank you so much for taking the time to actually break down the real science for us and get past the news headlines down to what the discoveries actually represent. And we're looking forward to passing that next stage of peer review and all of the future studies, hopefully crossing fingers that we have somebody out there who's going to fund the sort of more general population level version of this study as well.
Yeah. And I want to give a shout out to Dr. Michael Corley for allowing us to be on this study. It was real fun time analyzing the data, which is the nerdiest thing I could probably say. And then also the other authors on the study. The other one I wanted to shout out because you were talking about long living animals and the impact of aging is Vera Gorbanova, I believe from University of Buffalo. A lot of her work is actually looking at blue whales and all of these long living animals. Definitely check her science out. Actually, maybe even have her on the podcast. I think that she'd be a fantastic person to talk to.
Awesome. Thank you so much. I appreciate it. And looking forward to future conversations again.
Absolutely, Matt. Yeah, thank you.
Thank you.
Mm-hmm.